Good afternoon. Good afternoon. My name's Steve Alexander. I'm about to chair the session, which is slightly short notice, but we'll get through this. Is that a problem? It's a real pleasure to introduce Arthur. I've known him for a while. He's a long history of association with G protein-coupled receptors, which is the topic of his talk today. He was recruited to NCI by my predecessor, Michael Spedding, who was due to chair this session, and I'm sure he's on his way somewhere, but we'll get to him in a second. So most of the audience, I suspect, will know Arthur and will have come to his talks before. And one of the things you'll get from that is his enthusiasm for the topic. He really engages in that, and that's a mark of how he is. He's not just an enthusiasm for pharmacology and GPCRs and the maths associated with the understanding of interactions between ligands and targets. He's a big enthusiast for guitars as well. So if you had the opportunity to Zoom with him over the last couple of years, you'll have noticed all these guitars hanging up behind him. So if you want to get him off the topic of science and GPCRs, start talking to him about guitars and music as an alternative. So he is currently Dean of the Faculty of Pharmacy, big fun, at the Monash Institute of Pharmaceutical Sciences in Melbourne, which allegedly is the largest group of Greeks in the Southern Hemisphere, which explains his surname, Christopolous. And he's going to talk to us today about allosteric drug discovery. He's been really keen on this topic for a number of years, as many of you who will have read his papers will identify. And what he's trying to do now is convince us that he can take it from the theoretical to the practical. So Arthur, if you're willing to take the stage, please.
Thank you so much, Steve. I also realise now the secret to getting a good intro is actually not tell the chair that they're chair, and so they say extra nice things about you. It is wonderful to be here. Just as an aside, Melbourne does have the third largest Greek population outside of Athens and Salonika. Not at MIPS, but the city itself. So another reason to come to WCP 2026. It's wonderful to be here. I want to thank David, I want to thank Amrita, and of course I want to thank Steve and IUFAR and NCI UFAR. We had a long association, many, many years. It was 2007 when I first started. So a lot of fun memories, and it's been a long time since I've actually been able to travel, so it's good to be back, and hopefully some of what I have to say may be interesting, new, or at least keep us all awake, or me. I want to start by just disclosing Patrick and I are academic co-founders of Septerna, a GPCR biotech company, but that's got nothing to do with what I'm going to show you today. And I also do have a sponsored research agreement with Karuna Therapeutics, but that's academic only, and I am going to show you some of what we've done as part of that collaboration today as well. That's the obligatory GPCR slide. I don't really need to introduce these proteins to this audience, but I don't even have an arrest in there, do you notice? That's how old it is. But the point I want to make when I always start talking about GPCRs is how incredibly diverse the array of activators or ligands for these receptors are, given the relatively common structural orientation of the protein itself. And it kind of makes sense because these are the major conduit by which we sense our external environment and convert it into intracellular biological responses. The reason we can see, the reason we can smell, the reason we can taste, the reason my heart is racing, my mouth is dry, my stomach's all clammed up, it's because of GPCRs and the way they work. Of course, they're naturally allosteric in the sense that the activation of the receptor, depending on the binding locus, or irrespective of the binding locus, will interact with the transducer protein to initiate the response. But the key I wanted to make on this, of course, is the diverse array of activators. Nonetheless, just like all drug targets, despite the fact that GPCRs account for at least about 1/3 of the current medicinal drug targets, there is clinical attrition, and you've heard multiple talks already in this conference about are there problems with the targets, are there issues with mechanisms, are there issues with models, are there issues with trials, and the answer is, of course, you have issues with all of those, not just the other fact that all of diseases are polygenic in nature. But I think, from my perspective, one of the interesting things was the way we would often approach a target or approach, I'm using the word approach in more than one way, how we will tackle a therapeutic target. And if it doesn't seem to work, you'd move on to something else or something new. But I think there's a lot to be said, whether it's at the level of the target or any of these other things, that old targets and new approaches is what sort of got me interested in doing what I do. And you replace old with clinically validated, and you can understand where this could be going. And a new approach could be, can we target the target in a way that will actually get us to where we want to get there, especially if it's clinically validated. And that is how I view our approach to targeting these things allosterically. But there's another point I wanted to make, and it's about silos. It's interesting, when you think back, a lot of the successful GPCR drug discovery was led by pharmacology, and of course, it's medicinal chemistry programs. A lot of other drug target classes had structures way before we did, and therefore they would have a structural view or a structural advantage. And so you can look at it both ways. Now GPCRs are starting to come up with a lot of structures, and so that's informing what we're doing. But it's a reiteration to me that we should be looking beyond what we do in terms of our favourite, whether it's a favourite receptor, favourite family, favourite disease state or whatever, what's happening in other areas, because allostery is universal. It's always been around in other proteins, for example, hemoglobin, all the way back to Niels Bohr. We've got enzymes, we've got ion channels, okay? That's the work on the GABA-A receptor. And of course, the classic papers by Monash, Jacob, and the one where they actually came up with the actual model. The term itself, by the way, was actually coined in 1961 by Monash and Jacob at the end of one of the Cold Spring Harbor meetings. Allosteric. The other thing to point out about the classic paper here from Jean-Joe et al., there you go, that's what they use the term allosteric, but the primary binding site there, they refer to it as isosteric. And of course, isoster has other connotations in chemistry. So if anyone knows where the word allosteric actually came from, I'll give you, you get a prize. All right, this is what I'm going to talk about. The promise of allostery. Why do we like these things so much? Well, this is an analogy that many people have used, not just me. If you look at the endogenous binding site as an on-off switch for agonists or antagonists, competitive antagonists, molecularly, mechanism-wise, allosteric binding sites, just like the G protein binding site, act akin to dimmer switches. You can dial the activity to a predetermined level, either up or down, over and above which no more effect is observed in theory at maximal occupancy of the allosteric site. So we often talk about dimmer switch medicines these days. And if you think about what sort of advantages these types of dimmer switch medicines can offer, they're listed here. The most obvious one is the potential for greater subtype selectivity. And that can arise to a variety of mechanisms, and I'm going to come to those in a minute. But the traditional view has been that allosteric sites can be less conserved than orthosteric sites because there's not necessarily an endogenous ligand for that site. We can talk about that at the end of my presentation if you like. The second one is the titratable effect, the ceiling level to effect. That is really important. So that is where the dimmer switch mechanism comes in, where you can titrate the drug to the disease, and no matter how much more drug you put into the system, you're not going to get more of an effect either in a positive or a negative direction. That helps potential in, for example, on-target overdose. Linked to that is the one on the corner down here. There's no laser pointer, so I'm going to try and use my mouse. The quiescence in the absence of tone. If you get that, you can have a drug that basically sits there and doesn't do anything until the body itself decides when and where to release what it needs to release under both physiological and pathophysiological conditions. You put those two together and you can get some really, really nice modes of pharmacology that offer better translational potential. And finally, you can't talk about this if I don't mention bias in some way or another. You can have biased allosteric modulation, you can have biased agonism. Bias is simply just allostery directed into the cytosol. So the modulators are basically the transducer proteins and whatever's activating the receptor. It's all part and parcel of the same thing. It's interesting that I've got potential biased modulation / agonism because the story I'm going to finish with, I don't know which one of those it is at the moment, but it'll be, I'd value your opinion. Okay, so part one of my presentation, and that's going to be probably the largest part of the presentation, I want to talk about different ways that we've learned about how subtype selectivity actually is achieved by allosteric drugs. Part two of my presentation is linking those two molecular properties I described before that differentiate these dimmer switch medicines from orthosteric medicines, and it's at least a proof of concept story, a nice story of how you can really take advantage of an allosteric mechanism in a way that you can't achieve with an orthosteric mechanism. And the last part of my presentation will be about an interesting molecule that's we're still learning about now, even though I studied it a long time ago too. All right. Before I proceed, just a couple of words here. I've used this slide before. In an orthosteric world, however you want to quantify things, you need to be able to talk about how well something binds and how well something activates or inactivates. We and many others have used variants of the black left operational model to do this, because what you want, and you're always facing this issue. The issue is a trade-off between empiricism and mechanism. Ideally, you want to be able to quantify things as mechanistically as possible. You don't want to describe things as empirically as possible, say like an Emax, but somewhere in the middle is where you meet if you actually got a lot of drugs and a lot of assays and a lot of things and you want to progress a medicinal chemistry program. Jim Black showed us how well this could work. And what's happened more recently is we've realized you can use the same model based on affinities and efficacy measurements to quantify biased agonism. That's all I'm going to say on that. In an allosteric world, you got more numbers. And this is an important point because that's part of the discussion that you have with your medicinal chemistry colleagues in trying to drive programs forward. If you want an antagonist, how well it binds is probably enough. But if you want an allosteric modulator, you have to understand how it binds to the allosteric site, but you also have to understand and quantify how it modulates. And it can modulate affinity of orthosteric ligands, efficacy of orthosteric ligands. It can activate the receptor on its own, or that's a combination of all of those. So allostery needs more numbers. But operational models do allow us to get at those numbers. You can have a plethora of behaviours, but I'm only going to highlight the three obvious ones. The PAM is a positive allosteric modulator, so its cooperativity, its overall cooperativity is greater than 1. The NAM, the negative allosteric modulator, its cooperativity is less than 1, but it can go as close to, once it gets towards 0, it's indistinguishable from a competitive antagonist, unless it's killing efficacy, then it looks like a non-competitive. And the other one that we often tend to forget is the neutral allosteric ligand, or the NAL. And I think there's something to be said about NALs, and we may have an opportunity to talk about that later, but these are the ligands that don't change the properties of the orthosteric ligand at equilibrium, but can still occupy the allosteric site and, for example, block other allosteric modulators from binding to that site. In the GABA-A field, they know that very well, but they don't call them NALs, because they didn't read the NCI-UFA allosteric nomenclature document from 2014. All right, so part 1. That's for you, mate. No, it's a good thing. You're on the paper. So is Michael. So is Bill. So is Ral. That's okay. Oh, Michael's here. Sorry. All right. Okay. So let's start with the basics. As I said, the most obvious mechanism for allosteric ligand selectivity is that the dimmer switch region on one protein isn't there on another protein, or is in a different spot. Okay, non-conserved binding sites. And certainly, once the structural biology revolution began, we published this in Nature in 2018, and I used to get asked how many were the allosteric sites in GPCRs, and I would say everywhere. And then the structure started coming out, and guess what? They're everywhere. But please note the year, that was 2018 when this came out. And as of late last year, it's now at least 33 X-ray structures, 33 cryo-EM structures, which gives us at least 66 structures and more published of allosteric ligand-bound GPCRs. And boy, has that expanded that analogy. This is taken from a review from Dave and the team at MIPS. Class A, you can see the Class A receptors on the left, Class B receptors on the right, a whole range of regions where allosteric ligands can bind relative to where an orthosteric ligand binds. So yes, they can have different pockets, substantially different pockets, and vary between receptors. And there's your Class C, some Class C examples. So in one way, so actually the point I was trying to make here is that one receptor's allosteric site can be another receptor's, orthosteric site, and vice versa. Right, that's the plasticity of GPCRs and the way they functionally put together, structure put together, allows for that. We can talk more about whether these sites, there's endogenous allosteric ligands or not, but at a minimum, we should realize, knowing what we know about binding, that's why I started with all the diverse activators at the beginning of my presentation. One receptor's ortho site can be another type of receptor's allosteric site. But what have we learned since? There are other ways by which allosteric ligands can engender selectivity. And the first one I want to talk about is what we refer to as cooperativity-driven selectivity. That is, you could have an allosteric site that actually is more conserved than you thought it was across similar classes of a receptor subtype, but it only modulates at one subtype and doesn't do anything. It's a NAL at the other subtype. So it binds, but it doesn't modulate, and you can still get selectivity. And therefore, that selectivity is driven by the cooperativity, the communication between the allosteric site and the orthosteric site. Is there an example of that? Well, let's use my favorite receptor family, the muscarinic receptors. that have historically been notoriously difficult to target through the orthosteric site because it's completely conserved. But if you notice at the top here, these are all inactive state structures, but if you look at the top, there is a vestibular region, called the extracellular vestibule there, that's conserved across all 5 receptors, and we know that comprises an allosteric binding domain for these muscarinic receptors. So they all have an allosteric domain in the extracellular vestibule. So how are they selective? Well, let's have a look at this. I think I've shown some of this before, but we've got some new data that I want to talk about. Not published yet, still trying to work it out. Let's start with our favourite M4 PAM. This is LY2033298, was the first generation M4 PAM discovered by Eli Lilly. I was part of the collaboration with Chris Felder and the team. This was published in 2008, but it was actually discovered in 2000. That's how long sometimes it takes things to publish when you're working with drug companies that keep changing CEOs. What's happening here, essentially, this is a radioligand binding assay. We're using an orthosteric antagonist as the label. So what you're seeing is a drop in the binding of the antagonist, but a significant potentiation in the binding of the agonist. So it's a NAM of the antagonist radioligand, but it's a PAM, a very robust PAM, of acetylcholine. And you can see that the alpha factor, at least in this assay, is 290. With NMS, it's indistinguishable from competition, but it's a micromolar binder to the free allosteric site. Now, if you look at the other NM1 receptor, the same ligand hardly does anything. So it's essentially neutral with NMS, threefold at best with ACTH, so it may as well be a NAM, but it's also a micromolar binder and around the same affinity as the M4 PAM. So it's not the binding that's doing this, it's the cooperativity. Now, we can talk about the link between binding and cooperativity later. Let's do the reverse experiment. We'll take our favourite M1 PAM, not our favourite, actually the first generation M1 PAM that came out in 2009, I think, BQCA from Merck. That's the one we we all did a lot of work on earlier on because we didn't have anything else. But again, you can see a similar phenomenon. At the M1 receptor, it's an M1 PAM, it's a NAM with the antagonist, it's a robust PAM with the agonist. You put it on the M4 receptor and you see very little. These are selective ligands, no doubt about that. But they are still binding across more than one subtype. So one of the interesting things a few years ago was when we solved the structure of the M4 receptor. This is actually a model at the time of the PAM, the M4 PAM, bound to an acetylcholine-occupied receptor. And we had identified a number of residues between the orthosteric and the allosteric sites through mutagenesis that significantly affected the allosteric ligand affinity or its cooperativity. So there were 25 of these residues. Almost think of them as a network, but let's not. 25, 19 of those are conserved, 6 are not conserved. And we asked the question, how is there a transmission mechanism that involves this network in some sort of way? Because obviously, he's looking at it from the top, and these are the 6 residues that we identified that are non-conserved between the M1 and the M4 receptor. Now, some of them obviously point into the allosteric pocket, but some of the other ones, if you look at team 2, point straight out into the membrane. And as I said, there are others in between that are, albeit conserved, that also affect allosteric modulator properties. So we asked the question, is cooperativity, cooperativity-driven selectivity a real thing, and maybe it's mediated by some sort of network of a hotspot of amino acids? So we literally, as you can envisage, we just swapped all 6 to begin with and see what happens. And this is what happened. So at the M1 receptor, we take the M4 PAM, and as I showed you before, it's not doing anything. We take those 6 residues out of the M4 receptor and we put them into the M1 receptor, and the M4 PAM becomes an M1 PAM upon that 6 residue mutation. Now you know where this is going, right? The opposite experiment. There's the M4 receptor bound to an M1 PAM. We take those 6 residues out of the M1 receptor and put them into the M4, and we turn the M1 PAM into an M4 PAM. There's been a lot of work on this in the lab over the last couple of years, and you can envisage there's a lot of combinations of mutations and permutations that we have done as well. There's those 6. One thing we have found, and because I want to take up all your afternoon and evening, we narrowed it down to 4 residues that are necessary and sufficient to mediate this swap. That's conclusion number 1. Conclusion number 2, this phenomenon is conserved across multiple M1 and M4 PAM scaffolds. And that's what I'm showing you on this slide. So I've already shown you LY298, and I've already shown you BQCA. Now I'm showing you up the top there, emraclidine. That's a new M4 PAM that's currently just completed phase 1 clinical trials for psychosis. That's a Cerevel compound. And you can see at the M4 wild type, you get exactly what you would expect. It does nothing at the M1. now we've introduced what we call the quad mutations. I've defined them down the bottom, but there's 4 of those mutations from the M1 into the M4 and into the M1, sorry, and emraclidine becomes an M1 PAM. And down the bottom, we're starting with one of our own compounds, MIPS 1780. That's an M1 PAM that we developed in-house. Nothing at the M4 wild type. Swap those 4 amino acid residues and bang, there you go. So this seems to be a real phenomenon that goes across scaffolds. So cooperativity drives selectivity as much as binding site diversity perhaps drives selectivity, at least for certain types of receptors. So the major mechanism, of course, is conformational selection, but we do appear to have some sort of communication network going on between the allosteric and the allosteric binding sites. And what may be happening? Well, if you look at the top of the pocket at least, so I'll take it through. This is where it would have been good to have a laser pointer. I'll try to use a mouse. These are MD simulations done by Ron Draw for us at Stanford University in his lab. And essentially, if you just focus on the M1 receptor here, we quantified the distance between this cysteine here in the ECL2, it's 2.50, the ECL2 2.50, and this tyrosine, because what the MD showed, which we wouldn't find in any structure, is that this is flipping back and forth between these two, and if it's in this position, it creates a non-planar cryptic pocket, and the M1 PAMs are non-planar, and if it flips over to this position, it creates a planar pocket, and the M4 PAM scaffolds tend to be planar. And so what Ron did with the MD in his lab is they quantified how it was flipping the distance. So if it's in this orientation here, it's going to be a small number in terms of the angstroms, and if it's down here, it's going to be large, I should say. And if it goes here, it's going to be small because we're quantifying that and that. So if you're here, you start low, and each of these panels represent 3 different MD simulation runs. It's not so easy to see through all those colors. At the M1, it's done over 2,000 nanoseconds, so 2 microseconds. And what you're finding is this pocket is opening up a lot spontaneously, this cryptic pocket. So there's whenever acetylcholine, or in this case, notice acetylcholine is bound. And over here, we're just summarizing the findings. So there's basically a population of receptors that are in the non-M1 PAM preferring pocket, and there's a population of receptors that are in the M1 PAM preferring pocket, the cryptic pocket forms, and that's at the wild type receptor. Essentially, once we introduce those 4 mutations into the M1 receptor, we disfavor that type of cryptic pocket formation substantially. You can see now the tyrosine likes to hang around over here with this ECL2, it just stick in there, and therefore it creates a planar pocket, and all of a sudden they're preferentially recognizing M4 PAMs. And that's shown down here, where you can see not a lot of movement. In one of the simulations, you might see a bit of the opening of the pocket. Now over here is what we see at the M4 receptor with the similar sort of swap. The M4 tends to do this flipping and flopping over a much slower time scale. So that's why they took it out to 4,000, and you can see not much of activity because I don't have the figure here, but it would sort of look like that, the pocket, so it's not moving much. And eventually, if you look down here, once we put the mutation into the M4, the M1 residues into the M4, you do start seeing openings of this cryptic pocket to make it non-planar, so it favours M1 PAMs. So we think that's what's happening at the level of the pocket, but whether the whole network is required to transmit this mechanism, we still want to determine. And that's what's summarised over here. So basically, once you put the mutation in this M1 receptor with the quad, you're locking it almost essentially into an M4 preferring allosteric state. So there's something to be said about the potential for cooperativity networks or not. That leads naturally, I hope, to the whole idea of dynamics as well and how that contributes to allosteric drug selectivity, because when you're solving structures, it's fantastic, but you want to know more about the dynamic nature of these proteins because they are dynamic proteins. And so more and more we turn to that to complement our pharmacology, our structural biology, and anything else that we do when we're trying to understand these things. So to do that, I'll tell you a little bit of this story that we just published recently. This time we're focusing on a single receptor, the M4 receptor, okay? Just a single receptor, and we have some first-generation PAMs which bind to that allosteric site, but they do have distinct pharmacological properties. So on the left again is the L-white compound that I showed you earlier, the Lilly compound 298. This is more recent data. It's a micromolar binder. It's a good modulator. The agonist note is iperoxo. It's a high-affinity acetylcholine receptor agonist. It's not ACH in these experiments. It does signal, and you also see allosteric agonism, but that's probably overexpression more than anything else. On the right, we've got one of Vanderbilt's compounds from Craig Lindsley. Some of you may have seen this compound before in other presentations. So that's a much more modest binder. It's still, modest modulator. It binds in the same sort of affinity range, but it's got much weaker cooperativity. So the question is, what's going on here? You've got two allosteric modulators binding to the same pocket, similar affinity, but very different types of PAM pharmacology. There's a lot of work in this paper, so I'm just going to try and summarize just a couple of key findings. So, of course, we went and we solved the structures of these complexes. We've also solved the structure of the M4 bound to acetylcholine. So we've got on the left the M4 receptor. It's a heterotrimetic GI protein and iperoxo. That's the agonist-bound structure. In the middle, we've got the agonist plus the Vanderbilt PAM, and on the right, we've got the agonist plus the Lilly 298 PAM. You might be able to see them there, but that illustrates, especially if you look at the middle. Sorry, on the left, we're also showing you we do have ACH in the author structure as well. Look in the middle, they're overlapping in that classic extravestibular allosteric binding pocket. But why are they so different? So to do that, we turn to MD simulations again. This time, this was a collaboration with Ying-Long Miao and Kansas. And Ying-Long uses a technique called Gaussian accelerated MD. So the timescales are a little bit faster here to see what's going on with these drugs. So on the left, we start with the agonist-bound structure. In the middle, it's the agonist and the Lilly compound. And on the right, it's the Vanderbilt compound. And hopefully that shows that. Already, you should be able to see, this is one of these, you stare at it long enough and the dinosaur comes out. No, just look. You should be able to see differences. Just focus on the right is the agonist activating the G protein. The agonist G protein complex alone. Look at the Lilly compound in the middle up the top there. And then look at the Vanderbilt compound over there on the right in yellow. But I'm pretty sure visually you can see there are differences in the stability of these complexes. Now, of course, we're not just going to say, all right, we'll leave it there and they look like nice wiggly cartoons. Ying-Long did quantify these. What I'm showing here are 3 runs where he compared the MD poses in a modelled receptor to what we're seeing in the cryo-EM structure itself. And I think it's pretty obvious that the Vanderbilt compound, the weaker PAM, samples more conformations, is not as stable, it doesn't stabilise the complex as the Lilly compound. And so we do have dynamic mechanisms that contribute to how strongly an allosteric modulator is an allosteric modulator. So going forward, as I said at the outset, I don't think we can, we don't do structural studies without including dynamic studies, and of course pharmacology, because we love pharmacology and it starts and ends with pharmacology anyway. So these are the take-home messages. I've tried to get through this story. Site and sequence divergence. Okay, one GPCR type's ortho site can represent another potential allosteric site, depending on the class of GPCR. And they are everywhere. The overall mechanism is conformational selection, but there may be preferred cooperativity networks between orthosteric and allosteric sites that may contribute to the observed selectivity. Structural biology is indeed providing unprecedented insights into how these drugs work. But dynamics also play an important role in modulation and ligand selectivity, and you really can't uncouple one from the other. So it's a good time to be studying allostery again in the era of structural biology, but it's great to see that just solving a structure now is not enough, and we knew that anyway. All right, I'm going to move on to the second story. This one is a little bit more translational and a nice example, as I said, of how an allosteric drug can do something that an orthosteric drug can't for an unmet medical need. And the story I'm going to show you, we published it a couple of years ago, but I really like it and we're moving on with it and progressing it. And it's about chronic neuropathic pain, and it's about the adenosine A1 receptor. So this is a spinal cord, a dorsal horn of the spinal cord, and we know that adenosine is released by primary afferents because it's one of the body's most cytoprotective molecules. And in the spinal cord, adenosine release quietens things down, it dampens pain signals. And we know that, this is lamina 1 and lamina 2 under normal conditions. If you induce neuropathic pain, for example, the model that we use is partial nerve ligation, tie off one of the nerves, and within 2 weeks a rat develops mechanical allodynia. You get a significant enhancement, release of adenosine tone as part of the disease process, as part of the pain generation process, because the spinal cord is trying to say, shut the pain down, shut the signal down. So here, you have a disease context-specific condition and a tissue-specific condition where you can ask, can a drug work better in the disease context than it would if you didn't have the disease model, the disease state? Context-specific pharmacology. And that's where the dimmer switch analogy comes in, because allosteric interactions are reciprocal. What the PAM does to the agonist does to the PAM. Therefore, a PAM of adenosine should work better under the conditions of chronic pain than in the absence of chronic pain, because there's less adenosine tone there. So it's all about tone. So to do that, we started with a couple of compounds that we looked at a while ago. There are, Ken Jacobson knows this quite well. 2 amino-3-benzyl thiophene. The first one on the left, VCP-171. That's how old it is. VCP stands for Victorian College of Pharmacy. That's what we used to be called before we became Monash Institute of Pharmaceutical Sciences, i.e. MIPS. So you've got one on the left, one on the right. These were generated by my collaborator, Peter Scammell's head of medicinal chemistry at the time. So we've got these are 2 PAMs of the A1 receptor. The characterization you're seeing here is in a recombinant cell line, so you're getting potentiation, but you're also getting allosteric agonism. The key thing to note is they're micromolar binders, but their cooperativities are quite different. One's about 6, one's about 60 odd. So the dimmer switch setting is different for these 2 molecules. So the question then is, can we test our dimmer switch hypothesis in a disease model or a model of neuropathic pain? So we're collaborating with Wendy Imlach, who's a fantastic pain researcher at Monash University and a whiz electrophysiologist. Wendy did spinal cord slice electrophysiology, and for those of you that know about this sort of stuff, that is really hard to do. So she's really, really good at this stuff. So what we're showing here, so what Wendy would do, we would stimulate the primary I didn't tell you this. We'd stimulate the primary afferents and then measure evoked excitatory postsynaptic currents, either in lamina 1, the projection neurons, or in lamina 2 as well. So these are evoked excitatory postsynaptic currents, and adenosine tends to dampen that. So what you're seeing here is a baseline trace. VCP-171 and MIPS-521 both reduce those excitatory currents. That's the PAM effect, and they're blocked by DPCPX, a prototypical A1 antagonist. So it is A1 receptor mediated. For those of you that may ask, what about the allosteric agonism and all that, and you saw in the previous slide? Well, I'll tell you right now, it's not that, because in these preps, we've done separate experiments in a previous study where we treated the spinal cord slices with adenosine deaminase to kill all the endogenous adenosine, and these compounds don't work. They don't do anything. So they don't activate as allosteric agonists, and they need adenosine to show an effect. They're PAMs. They're pure PAMs in the spinal cord. Now, the interesting thing is when we moved on, hopefully you can see that data okay. I apologise, it should be a little bit bigger. Looking at the dose-dependent effects of the 2 PAMs in either sham-operated animals or after partial nerve ligation. So these is, again, EFIS recordings, but dose response. So if you look at the blue are the curves for VCP-171, the one with this lower positive cooperativity. As you increase the concentration of PAM, yes, you do see a reduction in the current because it's boosting whatever adenosine is there. But importantly, after partial nerve ligation, we've got a lot more adenosine. It has no further effect. It's no different to control, to vehicle. In contrast, MIPS-521, which is a stronger positively cooperative modulator, does have a significant difference between the sham-operated animals and the partial nerve ligation animal slices, suggesting that there is context-specific pharmacology happening here, and we've got enough positive cooperativity, enough of the dimmer switch setting to actually unmask it. Does it work? Well, if you then look at the animals in vivo, and this is intrathecal administration using the pull withdrawal, the von Frey assay, so there's the pull withdrawal effect. On the left, we hardly see anything with VCP-171 until we get to the highest possible dose we can give. But on the right, MIPS-521 gives a nice dose-dependent effect in reversing the allodynia. And so this is what I'm talking about. The beauty of this is, of course, we went ahead. So you've got to get the right dimmer switch setting, but once you do, you can have a drug that is going to work better in the disease than when you don't have the disease state, or it'll be more quiescent. And I can tell you, we've tested that. It doesn't have any effects on heart rate, because A1 receptors are important on heart rate. It doesn't have any effects like morphine does. We've done the rotor-rod assays and everything. So we hope that this type of approach can lead to a different class of, for instance, non-opioid analgesics for chronic pain. And to help us along that path, we solved the structure of the A1 receptor bound to both adenosine and MIPS 521, and that, you can see that in the paper there. And we identified a novel extrahelical allosteric site, and what we're doing now, using both traditional high-throughput screening, virtual ligand screening, and our internal medicinal ongoing chemistry programs, are trying to improve on the drug-like properties of these PAMs to literally see if we can come up with non-opioid analgesics that will work in the condition that you want them to work for chronic pain, which would be fantastic. So that's a nice example, I think, of the potential, the translational potential of allosteric drugs and the differentiation of allosteric mechanisms from orthosteric mechanisms. This is why adenosine agonists can do this too, but that you have way, way more problems with directly activating adenosine A1 receptors with an orthosteric agonist. So, this is the last part of my presentation, so we've got time to talk about anything you want. I remember, I'm not finished yet, it's okay, it's nothing. I just remembered, it was, I think, 2016 at the BPS. When I gave the Gatta Memorial lecture at the BPS in 2016, BPS meeting 2016, I remember at the time I showed a story, and this is more for the young people, the PhD students and the ECRs in the audience, where I said, I got my first paper was my PhD published in 1993 in the European Journal of Pharmacology. And 20 years later, in 2013, I got my first Nature paper on the same drug. So there's something to be said about perseverance. That's the story I told all the time. And I just realized, as I was putting this talk together, particularly this story, I did my postdoc in Minnesota after I did finish my PhD at the University of Minnesota, and then the Department of Psychiatry in 97, 98. I think I came back to Australia sometime during 99. And the first drug I studied as a postdoc in Minnesota was this compound over here called xanomeline. So it's interesting that some gifts just keep on giving. So I'm going to talk about this compound because I'm calling it the curious case of xanomeline because we're just starting to work out what's going on here. And for the first time in my career, when I've been referring to this compound, so I put orthosteric MACHR agonist up there, but I put inverted commas around the word orthosteric. In fact, I should tell you, I'll tell you, then I'll tell you this. I joined the lab in Minnesota to study the effect of the muscarinic nervous system, muscarinic receptor signaling in nitric oxide in the brain and in Alzheimer's disease. So that's kind of the reason I went to do that. And they gave me this particular drug to start with. Actually, they should give me this particular drug for real now after to calm down. Anyway, that's what we started. But the interesting thing about this is this compound has virtually identical, and when I mean virtually identical, it is almost totally identical affinities for all 5 muscarinic receptors in the orthosteric site. So its selectivity is driven purely by efficacy. And if you note down the bottom on the right-hand side, or left-hand side, Nature Chem Impress 2023. This drug's been around forever, and what I want to tell a lot of people, especially, again, the young people, is just because something's been around forever and it's in the clinic and everything, don't necessarily believe all of the experiments that probably should have been done were done to characterize it properly, and/or you can do it again anyway, and you should, because otherwise we can get in trouble for reasons you can imagine. And in fact, I remember I was telling, where's Alice? Alice over there, Alice Barizzi. I was telling her yesterday, Jim Black would do this all the time. The first thing that anyone that would join his lab, the first thing he would do would say, read something in the literature that you find interesting, and then repeat the experiment. Right? So sometimes we do a lot of dose response curves, so we do a lot of repeating of experiments, but I'll tell you what, you have to, and it's really, really important. And so it turns out no study had actually properly characterized xanomeline across all 5 subtypes in its common background ever. So it's always been pitched as an M1/M4 preferring agonist, but what we found is identical affinities, and it's way, way, way more M4 preferring than M1, and then the others, it's almost an antagonist of some of the others. So that's my little tangent. Not a rant, a tangent. Everything's a learning lesson. It's a learning lesson. A lesson. A teachable moment, if you will. Why were we studying this drug? Because at that stage, in 1997, when I started, it was actually just completed a phase 2 clinical trial by Eli Lilly in Alzheimer's disease patients. What they saw is it failed to meet its primary endpoint, which was around cognition, but they saw significant improvement in a cohort of patients, a subset in psychosis-related conditions. In fact, that's what then led them to do sponsor a small proof of concept trial in Anantha Shakar's lab. And he had about 20 schizophrenic patients, and they gave them xanomeline, and they saw improvements in positive, negative, and cognitive domains. Now I'm going to take a step over here and say, I don't know about you, I know there are other people that know a lot more about schizophrenia than I do, but pretty much every drug on the market treats the positive effects. You have to do a lot of convincing to tell me that they treat the negative, and in particular the cognitive deficits that are also linked to the schizophrenic syndrome. In fact, I don't think anything does. So having the potential to go cross over into those symptom domains is a big deal. Now, the other problem with xanomeline, one of the main reason it crashed out, was limited due to peripheral side effects, because it's either non-selective or it's an antagonist, so particularly the M2 and M3 was giving peripheral side effects. And even with the M1 in some cases, you can have bits in the heart and such. So what's happened since? Karuna, so Steve Paul, who was the CEO of Eli Lilly, our collaborator Chris Felder, who was at Eli Lilly, they got the company called Karuna, and they decided to go back to this and follow it up. And they took xanomeline and they went, again, back into the literature, and they found a very, very old anticholinergic called trospium. And the beauty of trospium is that it's peripherally restricted. So they just combined them, and they call it CAR-XT. And the idea being, of course, you're going to block the peripheral effects of xanomeline and hopefully benefit from the central effects of xanomeline. So that's where they are now. It's just completed phase 2 clinical trials. It's currently phase 3 clinical trials. I'm showing you the PANSS score data on the right there from the clinical trial. It's a really significant effect. This is the potential to be, if next year it comes through and gets FDA approved, this is the potential to be a new first-in-class therapy for schizophrenia in decades. And in our view, at least, it's primarily being driven by something going on at the M4. How the rest contribute, we don't know. But it's really exciting. But as I said, it's an old drug combined with an old drug to get a very interesting effect, at least proving that the M4 is worth going after, by the way. And I was saying it for years, but no one listened. No. And that's also, by the way, the reason why Lilly started an M4 PAM program back in 2000, because we'd already seen the xanomeline data, and that's what got them looking for M4 PAMs. But of course, the ones you're seeing now, the ones in clinical trials, is emraclidine, the Cerevel PAM, which is actually used to be Pfizer. Pfizer, all of them did the same thing. They all got out of neuropsychiatry just when I was saying to them, don't get out of neuropsychiatry. So Cerevel is actually Pfizer's neuropsychiatry. They spun it out into Cerevel, and the M4 PAM program went with it. And I just missed out. Same with Lilly, actually. Those compounds, they've got new compounds that's still on the shelf. I know about that, but no one else does. Anyway. Other similar scaffolds like xanomeline, though, also got into clinical trials, but they failed. So the question is, what's going on? The short answer I'm going to tell you right now is we're not really sure, but there's some interesting stuff happening. So the first thing we did, as I said, go back and actually do the proper experiments. Don't assume they've been done. So last year, an honor student in the lab with Celine Vallant and Dave Thaler, Jack McDonald, did this. We just profiled the signalling of the M4 receptor to xanomeline across a range of pathways, and of course, using acetylcholine as a reference ligand, doing the whole bias factor thing. We worked out that, I'm replotting the data on the left, that unlike acetylcholine, pilocarpine, well, not unlike acetylcholine, I shouldn't say that. Acetylcholine is a reference ligand. It has its own preferences. So we normalize everything to acetylcholine's preferences and to the GI2 in this case. But you can see that xanomeline at the M4 receptor has a different profile. It's a biased agonist at the M4 receptor. On the right, that's published data. On the right, it's unpublished. We've done that at the M1. It's not biased. We've done that at the M2. It's not biased. We're still doing it at the M3 and 4. We don't think, we're not getting, sorry, M3 and 5, and we're not getting a lot of signaling, but that's okay because it's saying it's an antagonist of those receptors, so that's telling you something. But the bias seems to be only at the M4 receptor, and as I mentioned at the very start of this section, this is M4 efficacy-driven ligand anyway, more so than anything else. So that's one thing. And the next bit of this story is, of course, we've solved the structure, and David's already presented this, I think, at the meeting on the weekend. And you think you've seen everything, and then you see something like this. Where does it bind? It binds to both sites. So it's the first time I've seen this on a GPCR. This is a late-stage clinical candidate binding to both the orthosteric and the allosteric site. 2 molecules, 1 receptor. We can talk about, in fact, I'm pretty sure we're going to talk about what this means later on. But that's what we saw. I'm just showing it there. And have a look. It really is sitting in the extracellular vestibule, recognised by those prototypical allosteric modulators, and what is, and as well as in the orthosteric site. And what's shown on the right-hand side in those panels, maybe just look at the very, very far right, where we've basically superimposed it on our PAM structures that I showed you in the previous section, the M4, the Lilly PAM, the Vanderbilt PAM, and even another Lilly PAM. It's recognising that pocket, and it's making a number of common interactions. Well, I should highlight in particular, if this works, remember this residue over here, phenylalanine 186. We've shown over and over that mutation, in fact, there's a, do I have it here? No, I don't. That's an important residue for the allosteric site, and knocking that out, which we're going to do in a minute, has an effect, a significant effect on a lot of these prototypical PAMs. So you've got to, that's why I said orthosteric stuff. I have no idea what this is. Right? I haven't come up with a name for it yet. Well, unless it's not, sorry. I know, I was going to say cooperative, but we're going to talk about that in a minute. We also collaborated with Ron Draw, and again, this is now using molecular dynamic simulations, and each of those figures there are 5 separate simulation runs, and the MD is looking at how often the allosteric site is occupied versus unoccupied. Ron started these simulations with the orthosteric xanomeline pre-bound, the rest of the drugs free in solution, go. And what you're seeing up the top there is xanomeline, in the middle, the typical classic PAM, the Lilly compound, and the bottom, the agonist, the high efficacy or high affinity agonistic peroxide. And what you're seeing is xanomeline is as happy as the classic PAM to occupy the allosteric site in the MD simulations, unlike eperoxo. A lot of orthosteric ligands do have to cross, well, all of them actually do have to go through that extracellular vestibule to get to the orthosteric site. And there's been prior studies in the field, many, led by Ron, showing that they can form metastable interactions in that vestibule before they finally settle down into their pocket. But what we're seeing here is something sees that pocket and tends to want to stay there. And I didn't expect that. And in terms of pharmacology, at the moment what we've done is we've used some dissociation kinetic experiments to see if we can get an allosteric effect manifesting. Now, the caveat here is that the radiolabel that we're using is an antagonist, tritiated N-methylscopolamine. But what you're seeing is dissociation mediated by isotopic dilution using 10 micromole atropine. And then we just added on top of that different concentrations of the PAM, the classic PAM. That's the blue curves up the top there, and they slow the dissociation rate, so there's an allosteric effect there. And as we increased the concentration of xanomeline, we also saw a significant slowing in the dissociation rate in the presence of xanomeline as well. When we mutated that phenylalanine residue I highlighted to you earlier, the whole effect is gone. So here we are. That's the paper. I stole this from Dave, actually. This is the paper, my first paper as a postdoc. And that was the figure that I actually had in the paper. It was at the M1 receptor. And we were studying the pharmacology because we thought there's something interesting about this compound. As was also the first time I ever got to use Adobe Illustrator, so I was really excited. I got to do a 3-dimensional box and everything. Look at it, fantastic, isn't it? I'm putting that there. We were studying a different phenomenon, but that was the figure from the paper in 1998, and it's just weird to have this structure now, and it's biased, and it's in clinical trials, and it's probably going to make it, and I don't know, but it's really interesting. Hang in there is what I'm trying to say. But I'd really would like to see obviously we're doing a lot of work trying to understand whether this mechanism is generalizable, whether it's specific to the receptor, but it's a really cool finding, I think. If it's not an artifact, which about half of you are going to sit there trying to ask me that question anyway, you're welcome to. I'm going to conclude with just a couple of words, just when you think this is why pharmacology is cool. Everything we do is driven by pharmacology. All right, so the question is always driven by pharmacology, by and large, at least for me, and then it takes you in whatever direction it has to take you, and you've got to do whatever you have to do to get at the answers. That's science. This is my favourite saying from Louis Pasteur. basically, if you make your own luck, and if you listen to your gut and it's usually right, just stop questioning it and just get on with it. So I'm pretty glad I stuck to the allosteric when a lot of people telling me not to. Here are a bunch of modulators that are currently in clinical trials. How many of those actually use some of the principles, especially the analytical principles that we have been proposing to be part of drug discovery and medicinal chemistry programs? I don't know. In fact, I know some of them haven't. That's why so many of them got rid of the psychiatry divisions, because they just thought bigger is better. No, bigger is not better. You want to understand the dimmer switch effect, and you want to dial the effect of the disease. So sometimes you need small cooperativity and for the disease. And the benzodiazepines and the GABA receptor have proven that. Their alpha factors are 3 to 5. You don't need 100. You'll kill a person. Anyway, it'll be interesting to see what comes from this. And the last thing, obviously, it's not just for GPCRs. These are universal principles. Just as I started at the beginning talking about silos, allosteric is universal. If it works on one protein, chances are you're going to see it on another protein. They're proteins. So look outside of your field. It's fun to do anyway. Sorry. These are my conclusions. You hit them all at once. I think it's clear now that allosteric sites are everywhere. The theory has become the reality. We do need a deep understanding of the mechanistic differentiators. This is vital for preclinical to clinical translation. We're not quite there yet, but we're getting there, and we have combining structural and computational biology with pharmacology, chemistry, more predictive models. There's a lot to learn, but it's fantastic to be able to bring all that together. Point 3 is the really, really important one for me in terms of translation, in terms of new types of medicines. Allosteric modulators can help for the potential to tailor the drug to the disease. That's the whole dimmer switch effect. It'll working in the disease and having minimal effects in the absence of a disease context. So context-specific, tone-specific, when I say tone, agonist tone, tissue-specific pharmacology. And for us, at least, analytical pharmacology underpins everything that we do, and the more we can incorporate it into our collaborations with medicinal chemists and such, as Jim Black always used to say, the dialogue between the biologist and the chemist is the most important thing you need to get right, and once you get it right, you can have a lot of fun. He didn't say that, that was the last bit was me. And on that note, I think I'll stop there, and I want to acknowledge these are the folks, I hope I got them all, that did the work that I showed you today. There are a lot of other really nice people working in our group doing different things. I want to particularly thank Celine Vallant and David Thal, the co-directors of our analytical structural neuropharmacology lab, that's what we're called. My long-term collaborators, my brother from another mother, Patrick Sexton, sitting over there. Give it up, Patrick. Woo! Peter Scammels is my cousin from another auntie. He's the chemist, so he gets second. Patrick gets first. Everyone in the team, former lab alum. I've mentioned Wendy, I've mentioned Ron, Ying Long. Andrew's been working with us for ages on a lot of this stuff. Craig, Rado for help with all the structural work. Jens and Nicholas were part of the A1 pain and the study. And they're working with us now on the virtual screening for new modulators that I talked about. And Chris and Steve from Karuna Therapeutics. I still have fun trying to explain to Steve Paul our own findings, even though he's on the paper, but it's great. I appreciate the support. And on that note, I thank you, and I'm happy to take any questions.
So, Michael, I think you got to the microphone first.
Yeah.
And then we'll
So,
go to Dan.
first of all, sorry I got here late, Arthur. I was communicating enthusiasm to young scientists, and it took longer to get across.
That's all right, mate. Us old scientists forgive you.
Oh, yeah, exactly. So I've got a question, because we were working on allosteric links with calcium antagonists a long time ago, and some of them were very temperature sensitive, and others weren't.
Right.
So how do you know if you're very temperature sensitive or not? Because it's obviously an energy dependent effect, and for some of the interactions, you could even reverse it depending on the temperature.
Well, to my knowledge, we haven't done that. Obviously, the temperatures that we're operating on are the sort of the standard. I mean, there's some work that's been done in terms of some GPCRs are voltage sensitive, which I don't know about that, but unless someone knows more than I do, we haven't done those experiments, so we haven't really looked for that, other than when we're trying to develop assays to look at, whether we can look at interactions over the long term, we play around with temperature. Having said that, when I think of those types of experiments, I haven't seen differences in the allosteric modulatory effect to suggest that we do have temperature to sensitivity, at least for the compounds that we've looked at.
Yeah, okay. No, but it's obviously beautiful.
Thank you. Thank you, Michael. Danny.
So some of the ergolines bind to both allosteric and autosteric sites, but of course it's different parts of the molecule. Now, xanomeline is an interesting beast. So what are the kinetics at the autosteric compared to the allosteric site? Can you do this?
Not yet. We are looking at, there are so many questions that are coming out. So we don't have any radio-labeled xanomeline. Trying to use a radio-labeled PAM to label. We can't knock out the orthosteric site yet. It's too hard because it's so conserved.
No, but you could have a negative,
actually
a neutral.
Yes. So let's jump to the chase. So this is my working hypothesis at the moment. The data that we've got around this molecule can be explained if it's a NAM, a negative allosteric modulator with the antagonist, and it's either a NAM or a neutral with itself. Yeah. And so that's, it's not a PAM. I don't think we don't have any interaction data that it's positively cooperative with itself. When you knock out the phenylalanine, which will then knock out the allosteric xanomeline, it's gone. Nothing changes. The affinity and the efficacy is purely driven then by the orthosteric. So it's either a NAL or a NAM with agonists, and it's a NAM with antagonists, and that explains the data because with the antagonist, it's. I know I've had this discussion with Steve, and I think it was asked of Dave. In the dissociation kinetic data, the potency of xanomeline to slow kinetics is 47 micromolar. Right? You never achieve that, right? But in the structures, we use 50 micromolar, and we've got a homogeneous population bound. Now, if it's negatively cooperative, 47 micromolar is, by the way, it's the affinity of the allosteric site for the NMS-occupied receptor. IC50 equals Kb divided by alpha. So if it's negatively cooperative, alpha is 0.1, 0.01. If you use that as even just a theoretical limit, the affinity of xanomeline for the allosteric site is actually better. It should be either 470 nanomolar potentially. It fits the classic ternary complex model, by the way, the data. But the now thing with agonists is yet to be, because the other hypothesis is it's either a NAM for both. If it's a NAM with itself, that means that dual occupancy, you're never going to really see in the brain. What you're going to get are two populations of receptor, allosteric bound or orthosteric bound. If that's true, that's interesting anyway. If it's a NAL, you're going to get them all bound, right? And it's not a PAM. That's currently where we're at.
Right. And I beat you. I started working on LSD in 1983, and I stopped for about 25 years, and now I start again. So.
Well, the LSD, I think it's still dissociating off the receptor from 25 years ago, right?
I'm still sitting there.
That's psychonetics.
Just before we go to Martin, is there anybody under 50 who wants to ask a question? I feel that we should be getting the younger...
I turned 50 in November. Does that count?
Yeah, we'll come to you in a second.
Yeah. You see, it was nice at the start. Now...
I'm 45, so... Hello, Arthur.
Hi.
Nice to see you again after so many years.
Yes, I'm trying to do that because obviously I'm blind. Yes.
Nice to see you again after so many years, mate.
Nice to be seen.
I've got a question that has two parts. So the first one is, do you think there are methods for high throughput screening for allosteric pharmacology to get all these parameters that you are looking at a high throughput mode? And the second part is, how much of the allosteric pharmacology do you think can drive drug repurposing instead of drug discovery?
Oh, two very different questions. Okay, the first one is yes and no in the sense that you can definitely do high throughput screening for allosteric modulators if you set up your conditions appropriately. But if you do say, for example, just simple titration mode, you'll get some composites of those numbers as initially. But, for instance, titration modes alone won't give you separate. You could get a KB or an alpha. You can get an idea, but it won't distinguish from an allosteric agonist, for instance. But that's the first bit of your funnel. The next bit is you can take representative molecules of those and do a more elaborate, nonetheless medium throughput pharmacology, and that's where you can get it still early in the medicinal chemistry campaign. In fact, I'd seen this so many times. When BMS, Merck, Pfizer, Lilly, and all of them, when they were having these types of programs, that's the problem they were facing. I could see what they were doing, but they weren't doing it right. At the time, everyone was basically going, bigger is better. We just want really high potency. That's what they did. They do the GABA experiment. You EC20, and you want it as big and as left shifted as possible. But that is actually a measure of the positive cooperativity with or without allosteric agonism. But no one thought about that, okay? So you're coming, if you characterize those sorts of molecules properly, you might get alpha factors of 100 or 1,000. They're going into animal models, and the things are salivating or dropping dead. I mean, I'm not kidding you. So you don't want to turn the agonist into a toxin. So now we know what we should have been doing. This is what I was telling them. When you do those initial titration, if you see things that look like a partial, what it means is they're a weak PAM. That's the compound you should be going after and seeing if you can optimize it, or you take one of each and you see if you can optimize them, because that is the way into a structure activity program that can tailor the drug to the disease. And I asked Pfizer before they spun out Cerevel, and I go, did you ever take your alpha 10s or alpha 30s, whatever? They go, no, we took all the hundreds and thousands, and they're the ones we progressed. And of course, all the clinical trials failed. They spun out Cerevel, and that happened with BMS, that happened with Merck, that happened with all of them. Pfizer, that's all what they did.
So last question from the under 50s.
Just. Very nice, Arthur. Thank you very much. Just this, my question stems a little bit from a conversation I had with Graham Milligan at the GRC, where he was giving me a hard time for studying allosterics on non-endogenous ligands. So going forward, do you anticipate an opportunity for dual therapy where you're putting in an agonist and an allosteric to, say, improve or reduce side effects?
Oh, sorry, yes, so the repurposing.
Or repurposing, for example?
We'll combine the answer.
Yeah.
Okay, yes. Yes and yes. And I think Graham's probably just stirring you up.
Yes.
Yeah.
Obviously.
For starters, right? Yeah, as long as you know about probe dependence, you're okay. Oh, that's the other interesting thing. We go back to the NAL. When we combine xanomeline with these prototypical PAMs, you get a very weak left shift as opposed. So now, if it's binding allosterically, we can explain why. It could be competing for the allosteric site. That's why the PAMs don't work as well. Combination therapy is, we've already called it a single target dual therapy approach. So that should tell you what we're doing now.
And so to follow up on that, you showed where you had the VU compound compared to the IX, and you had one, the allosteric was very stable, and you had a larger allosteric effect where the other wasn't. If you change the agonist, you talked about that dual communication between allosteric and orthosteric site. If you change the orthosteric agonist there, do you see the same effect, or I would assume it changes?
Yeah, so you're bringing in the potential for probe dependence there. At this stage, not for them, because we've done them against ACH, we've done them against that Ziparoxo that I was showing you, and that trend is retained. But for others, I just gave you an example of probe dependence with xanomeline, which may be competition for that. That's different again. Yeah. But that's a good question. Thank you.
Right, I think we're going to have to conclude there. So I hope you'll join me in thanking Arthur for not ranting and just lecturing us with his keynote today. Thank you very much.
Of a TCM-derived agent aimed at rejuvenating aging hematopoietic stem cells with predilection for lymphoid differentiation. Thank you, Dan.
Thank you, Chairman, for your introduction. Good afternoon, ladies and gentlemen. My name is Dan Gao. I'm from Xuanwu Hospital, Beijing in China. It's my honor to be here for this presentation. Today, my topic is identification of a traditional Chinese medicine-derived agent aimed at rejuvenating aging hematopoietic stem cell with predilection for lymphoid differentiation. As we all know, the problem of aging population becomes more serious. According to the nation's projection, we can see that by 2059, the proportion of 16 will reach about 25%, and the future will continue to be aging. So it's very important to improve aging and related disease. As reported, aging clock is regulated by many factors such as stem cell exhaustion, mitochondrial dysfunction, genomic instability, and telomere short, and so on. However, many experts believe that stem cell aging is the root of human aging. Actually, hematopoietic, here we called HSCs for short, occupied an extremely important place in stem cell, which has ability of cell renewal, proliferation, and differentiation into myeloid and lymphoid progenitor, leading to the balanced production of both myeloid and lymphoid lineage to maintain the homeostasis of blood lineage and the immune system of our body. However, with aging, HSCs result in abnormal self-renewal and differentiation, like decline in regeneration capacity, decrease in the lymphoid lineage, and increase in the myeloid lineage. Therefore, it's so significant to explore an intervention that can effectively improve HSC aging, especially lymphoid failure. Traditional Chinese medicine has a long history of anti-aging. As we see, Bian Qie, a famous doctor of Chinese medicine in ancient time, is said to live more than 300 years, and many Chinese herbs were recorded have anti-aging effects. So in this study, we first sequenced 9 active compounds from different herbs of anti-aging according to the clinical trials, reference, and ancient books in total body irradiated mice based on the CLPs and B cells, which was acute mouse model possessed aging HSCs phenotypes similar to naturally aging mice. We found that only one compound, TSG, could increase the number of CLPs and downstream B cells. TSG is one kind of steel-binding glucoside from He Shouwu, which has a long and profound history in prolonging life. Then we performed a comprehensive assessment on potency of HSC's rejuvenation in naturally aging mice. This is He Shouwu. In this study, 20 mice were treated with vehicle or TSG by gavage daily, which were analyzed after 30 days by FACS. It was found that mice that were treated with TSG presented a significantly higher number of CLPs in the bone marrow per mouse, and also great higher number of total B, pro-B, and pre-B of the B cells lineage compared to aging mass, but didn't produce any substantial effect on the number of T and myeloid cells of the bone marrow. Nevertheless, TS treatment results in significant elevation in the total number of 3 subset of T cells or SAMers and tend to restore balance as a large increase in the percentage of T cells and percentage of B cells and a decrease in the percentage of myeloid cells, but had no effect on the T cells in the peripheral blood. Moreover, TSG had no effect on the number of HSCs, including long-term HSCs and short-term HSCs, and common myeloid progenitors. We also detected ROS apoptosis proliferation in the LT or CLPs to investigate the potential cause of lymphoid increase after TSG treatment. We found that the percentage of BRDO positive CLPs, NAC5 positive CLPs that were unaltered in comparison to the aging mass treated with vehicle. Level of ROS was the same situation after TSG treatment. As we know, aging HSCs were also known to inhibit greatly reduced repopulation potential. Furthermore, we sorted HSCs or CLPs from different groups of mass and transplanted to the receptor mass to explore the influence on repopulation potential after TSG treatment. We found that the mice that received the long-term HSCs from the TSG-treated aging mass portrayed the higher contribution in all the cases, and the same phenomenon was observed in the CLP's transplantation. To explore the mechanism of TSG for anti-aging, we applied transcriptal analysis. It was found that the transcriptome of HSCs from the TSG-treated aging mice exhibited a profile analogous to that of the young mice. Gene set arrangement analysis and heat map analysis of 36 genes strongly tied to lymphoid lineage revealed that lymph-associated genes were significantly enriched in the TSG-treated aging mice. Some key genes were verified by RT-qPCR. Moreover, in the transcriptional analysis, some cell metabolism genes, such as oxphosphorelated genes and HSC's quiescent genes, were regulated by TSG. We can see that oxphosphor gene were down-regulated and the quiescent gene were up-regulated after TSG treatment in the LT of mice. Then we used seahorse to examine the metabolism of HSCs in the bone marrow. We can see that TSG could induce the production of ATP and keep the HSC state in the GO phase. Then we guessed MPK as the energy sensor, if it could be influenced by TSG to play the role. So we examined the protein expression by Western blot in HSCs. We found that the protein level of activated AMPK and its downstream of phosphorylate ACC were increased significantly, so TSG might maintain aging HSC's quiescence and activated AMPK. Then we treated mice with MPK inhibitor compensate by TL1 injection every other day. The results showed that compensate blocked the protective of TSG on the number of cell pieces and its downstream B cells in the bone marrow, as well as the proportion of B cells in the peripheral blood. For HSC transplantation, as shown in D and E, the percentage of donor-derived HSCs in the bone marrow of TSG-treated aging mice were also damaged with the treatment of compensate. Overall, the results suggest that TSG can effectively enhance HSC function and lymphoid differentiation, mainly through the activation of AMPK. Further, we also applied in vitro analysis to verify the mechanism of TSG for HSC aging via activating AMPK such as molecular docking, SPR, and promega enzyme activity assay. We found that TSG could bind well to AMPK. Maybe AMPK alpha 1 is a little better than AMPK alpha 2. The kinase assay was performed to measure luminescence-based AMPK activity. Clear activation of AMPK were triggered by TSG with dose-dependent manner. More importantly, we studied the effect of the TSG AMPK activity using specific AMPK inhibitor compensate and AMPK agonist ECA on synthetic cell lines. Using the Washington blot, we found that AMPK and ACC phosphorylation levels were ultimately increased in a dose-dependent manner after TSG treatment, which were even a little better than the ECA. Combined compensate with TSG, phosphorylation of MPK and ACC were dramatically decreased compared to TSG treatment. Taken together, these results indicate that TSG might promote MPK activation directly. Previous studies have shown that MPK activation is crucial to maintain the type 2 stability, and the type 2 as the one kind of demethylation enzyme was a substrate of MPK. Here we found that TSG treatment effectively boosted the protein level of active MPK and type 2, which were attenuated by the coordinated administration of MPK inhibitor compensate and TSG in the aging mice. We also found that in cell culture above mentioned. Furthermore, we evaluate genomic-wide DNA methylation of long-term stem cell isolated from the bone marrow of mice. Cluster analysis of the methylation profile revealed that the LT from the 4 groups fell into 2 categories: young and TSG groups clustered together, and old, as well as old and old TSG combined compensate group clarified into the same cluster. Specifically, the methylation level lymphoid regulated gene of the LT showed an evidence decrease in TSG treated aging mice, but were found elevated in TSG and compensate co-treated aging mice. To further validate this point, we adopt type 2 mutant mice and found that TSG treatment failed to increase the absolute number of cell pieces in total body irradiated type 2 mutant mice. We next verify the role of TAT2 by co-culture assay in vitro. The results show that HSCs sorted from the bone marrow of mice exhibit enhanced differentiation potential to lymphoid cells after TSG treatment, while TAT2 inhibitor Bobcat 339 significantly decreased lymphoid differentiation potential when co-culture with the OP9 strong cells. All above implies that TAT2 as a substrate of MPK has an imperative role in driving HSC differentiation towards lymphoid lineage. Collectively, in this study, we try to devise a new system for assessing a potential of individual agents for rejuvenating agents, HSCs. We successfully identified the compound TSG that exhibit potent efficacy in rejuvenating aging HSCs in aging mice and total body irradiate induced premature senescence. We deemed the MPK TAT2 acts as a potential mechanism of facilitating describing the metabolism to epigenetic signaling pathway for rejuvenating aging HSCs. That's all. Thank you. Thank you to all the audience, to my tutors and my teammates. Thank you.
Thank you for that. The paper is open for just one question in view of the time. If there is no question, then we move to the next speaker. Thank you, Dr. Gao, again.
Thank you.
Is Dr. Hilary Makiopas in the room?
Yes.
We welcome you, Dr. Makiopas. He will present us the paper TRPA1 as a potential factor and drug target in scleroderma. Dermal fibrosis and alternative macrophages activation are attenuated in TRPA1 deficient mice in experimentally induced scleroderma.
Thank you for the introduction, and good afternoon, everyone. I'm very humbled to have this opportunity to talk here in this great Congress today. And my name is Hilary Makiopas. I'm a physician from Tampere, Finland, and I'm doing my PhD thesis in the immunopharmacology research group led by Professor Eeva Moilanen in Tampere University. So my talk's title is TRPA1 as a potential factor and drug target in scleroderma. Dermal fibrosis and alternative macrophage activation are attenuated in TRPA1 deficient mice in experimentally induced scleroderma, so quite a long one. And there, under the title, are the names of other writers of this work. First, a brief background of the three main subjects that this work crosses over. First, on the left, scleroderma. It is a fibrotic skin manifestation of systemic sclerosis, which is a fibrosing rheumatoid disease affecting 1 to 4 people in 10,000. There in the middle, macrophages. Macrophages are known to have this spectrum of different phenotypes, two opposite ends of which are the classical activation, abbreviated M1 most commonly, and it's the pro-inflammatory phenotype of macrophages. The other end are alternatively activated macrophages, M2 macrophages that are most famously anti-inflammatory, but they are also remarkable in resolution and wound healing, and their kind of overactivation might lead to unwanted effects like fibrosis. And it is known that in pathogenesis of scleroderma, M2 macrophage activation is included. Then on the right, the TRPA1 receptor transient receptor potential anchorin 1. It's a non-selective cation channel, activation of which causes infuse of calcium ions, and that causes pain and release of inflammatory mediators. It is originally known to be expressed in neurons, but now more and more papers showing its expression in other non-neuronal cells as well as like skin cells and inflammatory cells. So we now know it's not only neuronal channel. So how do we wrap up these three subjects together to make a hypothesis? Earlier studies have shown that TRPA1 deficiency has an impact on models of M1-driven diseases like atherosclerosis and hypoxia-reperfusion injury. Then, as I said earlier, systemic sclerosis is an M2-driven disease, as shown in this figure on the right-hand side. It is also interesting that TRPA1 has been shown to mediate Th2-driven inflammation, which is closely related to the M2 macrophages. So, we hypothesized that TRPA1 deficient mice should have attenuated manifestations in experimental models of scleroderma. So, we first sent out to study the role of TRPA1 in experimental model of scleroderma in vivo, and for that we used bleomycin-induced scleroderma in wild-type and TRPA1 deficient mice. Then we wanted to investigate the underlying M2 macrophage-mediated mechanism in vitro, and there we used J774 mouse macrophages, cultured and commercially available. And then we also harvested mouse peritoneal macrophages from the wild type and TRPA1 deficient mice from our own lab. And now on to the results. From now on, you can see on the upper left corner, there's a box indicating what the current research subject was. So we found that the TRPA1 deficient mice have attenuated bleomycin-induced dermal thickening. So on the left here, we can see the demonstrative histology cuts, HE colored cuts of mouse skin that is bleomycin injected on the bottom side of wild type and TRPA1 deficient mice, and here are the control counterparts of both genotypes. As here, we can see the bars presenting the mean skin thickness. We can see that in the bleomycin-treated mice, the TRPA1 deficient mice skin didn't get thicker as much, so there's some form of downregulation there. Second finding was that bleomycin-induced collagen accumulation is attenuated in TRPA1 deficient mice. Here again, demonstrative histology cuts, but now colored differently with Masson's trichrome staining that highlights collagen. And here, these bars show means that indicates the collagen density in the skin, and we can see that the TRPA1 deficient mice have skin less dense in collagen compared to the wild-type animals. We also took the skin samples and extracted messenger RNA from them and ran RT-qPCR to see expression of 3 of the most common collagen types in skin, and we can see that also in mRNA level, the expression of collagen is downregulated in TRPA1 deficient animals. Here, we show that bleomycin-induced expression of profibrotic mediators is also lowered in TRPA1 deficient mice. So these are same skin samples as previously with the collagen pictures. There are many profibrotic markers here, and then also a couple of more classic inflammatory mediators like interleukin-6 and cyclooxygenase 2. The profibrotic factors on the upper row, as you can see, show that the TRPA1 deficient animals had lower expression of these profibrotic factors. Then, next finding, markers of M2 type macrophage activation are lower in bleomycin-treated TRPA1 deficient mice. So here we can see these are the two two main markers of M2 macrophage activation. In mice, I have to say, it's a bit different in humans, but in mice, these two are the main ones. And then interleukin-13, which of course belongs to Th2 lymphocyte or like inflammation family. So we can see that these M2 type macrophage markers were substantially downregulated in TRPA1 deficient animals, and also was the Th2 related interleukin-13. Now on to the in vitro results. Cultured J774 mouse macrophages were incubated in bleomycin in absence and presence of interleukin-4, which is a main cytokine of Th2-mediated immunity and a main inducer of M2 macrophage polarity. For the first time, we saw that bleomycin by itself dose-dependently increased the expression of M2 markers, arginase 1 and MRC1, as seen in these white bars over here. And when interleukin-4 was present, bleomycin greatly enhanced the effects of IL-4, suggesting an interaction, as seen in these gray bars over here. In this picture, on the left, there is a sum-up figure of intracellular mechanisms of macrophage polarization. M2 mechanisms are on the green side on the bottom here, and two of the main intracellular mechanisms of M2 polarization are phosphorylation of STAT6 protein and expression of PPAR gamma. From the J774 cultured mouse macrophages, we studied these two things, and here we can see that in case of STAT6, there is no difference. The bleomycin makes no difference here, but to the PPAR gamma expression measured as mRNA, there is the bleomycin somewhat boosts the effect of interleukin-4, which is best seen at 4-hour time point. To see if TRPA1 got anything to do with the M2 promoting effects of bleomycin, we harvested macrophages from the peritoneum of wild-type and TRPA1 deficient mice, cultured them and incubated with bleomycin and interleukin-4. We saw that interesting responses to IL-4 alone were significantly downregulated in TRPA1 deficient mice regards to M2 markers arginase 1 and MRC1, and also Th2 interleukin-13. With the M2 markers, we can also see that the marker expression boosting effect of bleomycin is somehow TRPA1 mediated, as you can see by comparing these two black bars and these two black bars over here. So finally, to conclude, these results show that TRPA1 deficiency downregulates bleomycin-induced processes like skin fibrosis, collagen accumulation, M2 macrophage activation. Bleomycin has intrinsic effects on macrophage polarization that are TRPA1 dependent, and therefore we think that TRPA1 antagonists could potentially be future drugs used in M2 macrophage-driven diseases like systemic sclerosis. And finally, I want to thank all the funders as well as all my colleagues at the Immunopharmacology Research Group. If you're interested, the article containing this data was published between the abstract submission and this Congress, so if you want to take a look at this open access article, there's the QR code you can scan. Thank you very much for your attention.
Thank you very much, Dr. Maki Opas, for your presentation. The paper is open for questions. I have one. In your model of scleroderma, have you characterized any agonist of the TRPA1 receptor that could justify the involvement of this receptor and the response to bleomycin?
So do you mean if we use any TRPA1 agonists in the model?
No, if there is, when you induce scleroderma in your mice by bleomycin, if you have the increase of some endogenous agonist of this receptor that could explain why the knockout mice for these receptors have a better response.
Yeah, we haven't done any analysis like that to like any oxygen or nitrogen species or any other endogenous agonists. We haven't done it, but that's a good idea and for the future. Yes.
Okay. Well, if there is no further questions, we thank you very much for your presentation. And I'll call now the next speaker is Dr. Elisa Porchietto from the University of Camerino. Is she in the room?
I'm here.
Oh, sorry. And she will present the paper Tetrabromobenzotriazole counteracts metabolic derangements evoked by the consumption of hypercaloric diet in mice.
Good afternoon, everyone. I thank the organizing committee for the opportunity to present here my activity research. During the last year, we have witnessed a lifestyle modification characterized by the increased consumption of unhealthy food and reduced physical activity that have spread the insurgence of metabolic disorders such as obesity and type 2 diabetes. This pathological condition share the common feature of a low-grade chronic inflammation, also known as metaflammation, that is becoming a new interesting field of research for counteracting metabolic disorders. Growing body of evidence suggests the role of kinases as crucial mediator for the activation of inflammatory cascades. As we could see from this graph, the correlation between kinase and obesity and kinase and type 2 diabetes has been increasing over the last 40 years, especially during the last decades. In the light of this, several small molecule kinase inhibitors were developed, and nowadays around 90 small molecules have been approved by FDA. The laboratory of Professor Massimo Collino from the University of Turin has already been proved how targeting inflammatory cascades is effective in ameliorating meta-inflammation. In these two works, they demonstrate how the high-fat diet consumption evoked the overexpression and the overactivation of NLRP3 inflammasome complex and JAK2 kinase, and how the pharmacological inhibition of these two targets is effective in reducing glucose intolerance and the higher level of pro-inflammatory cytokine in the serum of high-fat diet fed mice. In the light of this, it is well known how these inflammatory cascades are specifically activated by post-transcriptional modifications such as phosphorylation, which are mediated by kinases. Among them, we identify a crucial serine kinase named casein kinase 2, whose pathological role has already been proved in several pathological contexts. In particular, casein kinase 2 is directly involved in the activation of two well-known pro-inflammatory cascades, NF-κB and JAK-STAT, involved in meta-inflammation. For this reason, we selected a potent CK2 inhibitor named 4567 tetrabromobenzotriazole, or TBB, and assessed its effectiveness in a murine model of high-fat diet consumption. In detail, mice were randomly divided in 3 groups. One group was fed with a controlled diet for 12 weeks, while the other one with an high-fat, high-sugar diet. A subgroup of the high-fat, high-sugar fed mice received TBB at the dose of 2.5 milligram per kilo for the last 8 weeks of the protocol. Despite not being effective in reducing body weight gain evoked by high-fat diet consumption, TBB improved diet-induced glucose intolerance, as we could see from this data, representing a reduced fasting blood glucose level compared to the high-fat, high-sugar fed mice, and a lower area under the curve following oral glucose tolerance test. Moreover, TBB administration ameliorates lipid plasmatic profile. As we could see, both triglyceride and total cholesterol level were dramatically reduced compared to the high-fat, high-sugar fed mice. Next, we evaluated some biological marker of organ damage, such as the transaminase. Both AST and HALT level were massively increased in the high-fat, high-sugar fed mice, and TBB present lower levels in the plasma of the TBB-treated mice. Next, we want to evaluate the TBB effect in alleviating local inflammation at hepatic level. First of all, we evaluated the expression of a well-known adhesion molecule, ICAM1, which is known for recruiting immune cell in the inflamed tissues. As we could see, ICAM1 was overexpressed in the high-fat, high-sugar-fed mice, while TBB-treated mice presented lower expression of this molecule. Next, we want to characterize the immune subpopulation that was recruited in the hepatic tissue, and by the evaluation of mieloperoxidase, which is an enzyme involved in the neutrophilic infiltration. Both the expression and the activity of this enzyme was statistically significant reduced in TBB-treated mice compared to the high-fat, high-sugar group. Subsequently, we evaluated the drug target, CK2, in the hepatic tissue. As we could see, the high-fat, high-sugar diet evoked a massive increase of the catalytic subunit alpha, which is responsible of the phosphorylation of the target, compared to the treated mice. Next, we evaluated the role of the kinase in the crosstalk with the well-known pro-inflammatory signaling cascades such as JAK-STAT and NF-κB, and the potential beneficial role of TBB administration. First of all, we evaluated how TBB could affect the NF-κB signaling pathway. As we could see from this densitometrical analysis, the high-fat, high-sugar diet evoked an overactivation of both IKK and IKB kinase, along with an increased nuclear translocation of P65 subunit. TBB group, the TBB-treated mice presented a statistically significant reduced overactivation of both IKK, IKB, and a lower nuclear retention of P65 subunit. This data was further confirmed by the mRNA expression of TNF-alpha that was dramatically increased in the high-fat, high-sugar-fed mice compared to the treated one. Next, knowing the direct role of NF-κB in promoting the genetic expression of NLRP3, we evaluate whether the pharmacological inhibition of CK2 could be effective in reducing the overactivation and the overexpression of NLRP3. As we could see from this data, both NLRP3 and cleaved caspase dramatically increased by the high-fat diet consumption were statistically significantly reduced in the TBB-treated mice. In parallel, we evaluated the interleukin-1 beta mRNA expression, and as we could see, the TBB treatment was effective in reducing the diet-induced increase of this pro-inflammatory cytokine. In conclusion, we evaluated the effect of TBB administration on JAK-STAT pathway. We confirmed that this pathway is overactivated by the hypercaloric diet consumption, and that the CK2 inhibition was effective in reducing the overactivation of these pro-inflammatory cascades. This data was further strengthened by the evaluation of mRNA expression of interleukin-6, which is known for promoting the activation of these signaling cascades. And again, the TBB treatment was effective in reducing interleukin-6 mRNA expression. the reduction of local inflammation was parallel by a reduction of inflammation at a systemic level. These are the results of pro-inflammatory cytokine in the plasma of the mice, and as we could see from this data, the high fatty sugar consumption evoked an increase of all the pro-inflammatory cytokine that TBB was able to counteract. In conclusion, we could see how it is possible to targeting meta-inflammation, identifying a kinase, casein kinase 2, which is directly involved in the activation of other well-known pro-inflammatory signaling cascades, and how the TBB administration was effective in ameliorating the diet-induced metabolic disorder. In conclusion, I want to thank Professor Massimo Collino from the University of Turin, Professor Carlo Cifani from the University of Camerino, my colleagues for the support, and thank you for the attention.
Thank you, Dr. Porchietto. And the paper is open for questions.
My name is Allosteric, I'm Geneva University. It's a very simple structure, and the affinity to the CB enzyme is not very high, I guess. So you have used fairly high doses. Is there no better, maybe selective inhibitors of casein kinase?
There are many other inhibitors of CK2. It depends on their action. I selected a CK2 inhibitor, he's an ATP competitive inhibitor, according to ATP,
course, yeah,
yes.
Yeah.
And the dosage was selected according to previous in vivo study that using chronic model this molecule.
Yes, so it probably blocks other kinases as well.
It's pretty selective,
Yeah.
but it could be that we selected one of the most potent one that we could find.
Yeah. The first slide you showed on the increase of publications on kinase inhibitors and on metabolic syndrome.
Yeah.
There was a correlation. That was not this type of kinase. That was
no,
not
just
tyrosine
general kinase. Yeah.
Okay. Yeah. Good.
Yeah.
Thank you. It's a very well structured and nice work.
Thank you.
Yeah.
Further questions? I have one. Have you checked the contribution of other lipids on this model, such as cholesterol or triglycerides, and the effects of your CK2 inhibitor?
Thank you for the question. We just evaluate the total cholesterol and triglyceride level at serum level, but we not evaluate the signaling pathway correlated to cholesterol synthesis or triglycerides.
And it was lower?
Yeah.
Okay.
Yeah.
Any further questions? Okay, thank you very much for your presentation. And I'd like now to introduce next speaker. Sorry, I have to wait for next speaker is Dr. Zhang Yingming from the Huazhong University of Science and Technology, who's presenting the paper Platelet GP6 is a mediator of rhabdomyolysis-induced acute kidney injury in mice. Thank you, Doctor.
Thanks, Chair. Good afternoon, everyone. Today, my presentation is platelet GP6 is mediator of rhabdomyolysis-induced acute kidney injury in mice. I'm Zhang Yiming from Tongji Medical College, Wuhan, China. Acute kidney injury, AKI, its characteristics is high incidence, it's high mortality, and high hospital costs, and poor recovery. Because of low effective treatment now, so AKI is getting more and more attention from world. Rhabdomyolysis-induced AKI, it is RMAKI, is induced by muscular damage and rhabdolysis release myoglobin, introduce intratumoral obstruction. Myoglobin is also in the NO scabbering, lipid peroxidation, apoptosis, NF-kappa B, and TGF-beta release. All of this needs vasoconstriction, oxidative stressors, inflammation, and fibrosis. As you know, platelet is the smallest cell in the blood. It is resting form under normal condition. But when the vascular damaged, the platelet activated and involved in hemostasis and thrombosis. Beyond this, it's also involved in other disease pathology, such as inflammation, injury, cancer, and so on. In the inflammation, platelet interaction with neutral cells. Neutral cells, monocytes, as this interaction promote inflammation. And between platelet and leukocyte interaction have direct and indirect interactions. The direct interaction involves platelets receptor and the leukocyte receptor, the molecular pair, such as P-selectin on platelet and the PSGR1 in leukocyte. Besides this, other receptors also involved it. Besides this, after platelet activation, some soluble factors, such as soluble CD40 ligand and other chemokines, cytokines, and indirect make platelet-leukocyte interaction. And the GP6 is a platelet and macrocyte receptor, only expressed in platelet and macrocyte. It have extracellular domain, transmembrane domain, and cytometric domain. When the collagen, the GP6 ligand, binding GP6 and trigger the signal transduction, make the Fc receptor gamma chain item phosphorylation and signal transduction, make calcium increased and platelet activation and release. Have reported platelet GP6 is involved in many diseases, such as phenolmier-derived sepsis, myocardial ischemic peripheral injury, cardiac inflammation, the glomerulus, and cirrhosis. So, our project aims is to check whether platelet GP6 is involved in RMAKI and explore how does heming contribute to platelet activation and RMAKI, and check if blockade of GP6 elevate RMAKI. First, we histologically the RMAKI mice model and check the biological parameters, BUN and the creatine increased. This implied the model is done. And we find in the RMAKI model have some platelet in the initial and in the tubular cells. So this implies platelets participate in the RMACI. And then we checked the model mice platelet function and found in model mice the platelet function, collagen-induced and ADP thrombin also increased compared with control. And the granule release is also enhanced. Other functions is clot retraction is enhanced also. And in model mice platelet, the Jola-I binding and the P-selectin dispersion also increased. This implies there are some substances in the blood of model mice where active platelets. And the hemi is from hemolysis or tissue injury damage from hemoglobin or microglobin. It's hemi. Besides its effect in other cells, it also involved in some disease, for example, the ACI. So we check if hemi induced platelet accretion. We use in vitro experiment, dose-dependent hemi induced platelet aggregation and release. In vitro experiment, hemi also increased Jola-I binding and the P-selectin expression in platelets, in washed platelets. So this implies hemi active platelets and directly induced platelet aggregation and release. But why? We found in RMACI mice, GP6 expression increased and enhanced the downstream signaling. We can say, compared with control, GP6 increased. This is the total protein. And also, but other receptors like alpha-2B and P2Y12, no difference. And the downstream signaling, 6-phosphorylation, is also increased. So the hemi may be not only increase level of platelet GP6, but also increase the downstream signaling. So we use different hemi, check the watch the platelet signaling from GP6 downstream. We found the PRC gamma 2 and SIC also increased by the hemi. And in the watch platelet function aggregation, we can find like hemi, like the collagen, increased the aggregation. But we use GP6 inhibitor, JAK1, like collagen. It block hemi-induced aggregation. So this implies hemi activates platelet via GP6 and enhancing the downstream signaling. And in vitro experiment, hemi also increased the platelet GP6, but to alpha-2B, P2Y12 other receptors, no difference. So we can say hemi specifically increased platelet GP6. And we use experiment in vivo experiment. JAK1, day 1, this is a GP6 inhibitor, and day 4, make model, and day 5, harvest blood and candidates, we find the chemical BUN and creatinine decreased after inhibitor GP6. And from the hematologic, we found after use JAK1, GP6 inhibitor, the tubular injury score also decreased. So platelet GP6 promotes injury in RMAKI mice, and blocked GP6 can elevate kidney damage. Macrophage X traps, like neutrophil, also extracellular traps. Macrophage, when damaged, and platelet activation, and used with AKI, the macrophage DNA and histone released and makes mats. So we check GP6, if GP6 contribute to the mass formation. Compare with the control, inhibit JK1, inhibit GP6 with JK1, and decreased mass formation. And we compare with resin platelets. Resin platelets activation, the mass increased. But when you use GP6 blocker, the mass decreased. So platelet GP6 contribute to the formation of mats. In the summary, rhabdomyolysis and induce hemi release, and hemi active platelet from GP6 and make macrophage activation and forming mats, make tubular injury and induce the AKI. When use GP6 antibody JAK1 block GP6 and inhibit mass formation and elevate AKI. So this also thanks the Division of Nephrology, Tongji Hospital, Professor Zhen Rui. Thanks.
Thank you, Dr. Ming. And the paper is open for discussion.
Hi, very excellent job. I know your research field focus on planet, and planet can secrete TGF-beta, and it can promote cancer metastasis. So how do you do related work on that part? And platelet secrete TGF-beta also promote tumor metastasis.
Thank you very much.
Yes.
It's a good question. Yeah, platelet can release TGF-beta, and it is maybe 50% in the blood. So it is also promote cancer metastasis. Our lab will submission a paper about this.
Yeah. Yes.
Thank you.
Yes. Thank you.
Steve Watson, University of Birmingham. That was a very impressive presentation, a lot of data.
Thank you.
The demonstration for involvement of GP6 is very clear, and JAK1 is very specific to GP6, but heme has also been shown to activate platelets through the C-type lectin receptor CLEC2. How do you think that is contributing here?
You mean the CLEC2 is also involved in this process?
There are two papers showing that heme is a selective ligand for CLEC2 on platelets, so it is also activating.
Yeah, I think so. Besides GP6 and another receptor, CLEC2 also involved in the process. Have a paper have published. This paper is done 2019 before the Pacific COVID-19. So, but the COVID-19 has delayed the publication.
Okay. And then just a second question. You showed an increase on GP6 expression on platelets. What was the time course of that increase? How long did it take before the GP6 levels were increased?
Sorry?
I think you showed midway in the talk, if I followed you correctly, that the levels of GP6 on platelets in the model increased,
Yeah.
not only the function but the level. How long did it take for that increase in GP6? How many days upon the induction of injury?
In vitro experiment?
Yeah.
Yeah, in vitro experiment, we incubate hemi with 6 hours, and 6 hours is increased, maybe last, I'm not sure how long, but 6 hours is also increased.
Okay, I think we can leave it there. I'm not sure, since platelets aren't making new protein, it's not on intracellular sites. It looks as if it's somehow becoming more accessible, but I'm not sure how you can see such a change in that time. But very, very nice work. Thank you.
Okay, thank you.
Hi, I'm Steve McWilliam. I'm a senior lecturer from Liverpool.
Okay.
One of the other proposed mechanisms for the kidney damage in rhabdomyolysis is direct sort of damage of the heme or iron on proximal tubule cells, so it gets filtered and then it has a direct impact. Do you think that there are maybe multiple factors going on causing the kidney damage, or do you think that the mechanism you're proposing is the main one?
Yeah, but AKI, different model, different mechanisms. We just use the rhabdomyolysis-induced AKI. This is maybe hemi is the main point.
Which brings me on to my other question. So we see AKI much more commonly, or rhabdomyolysis is quite rare in our patients. The most common cause of AKI is post-cardiac bypass surgery, and we're beginning to think that maybe there's more of a role of hemolysis rather than just ischemia reperfusion in those patients. Have you done any work looking at the role of hemolysis itself and AKI?
Yeah, maybe ischemia is more common. Ischemia reparation induce the AKI.
Yeah, so we're just wondering whether there's a similar mechanism between hemolysis in the blood and rhabdomyolysis. I don't know if you've got any thoughts on that.
Yeah, in the ischemia and reparation AKI, platelets are also involved in that, but it's different mechanisms. Maybe has a paper 2 years or 3 years ago published. Yeah.
Okay. Thank you.
Thank you.
Thank you.
Any other question? I would like just one more question to ask, which is, have you measured the concentration of heme that you have in your model of rhabdomyolysis, just to be sure that it's in the same range of the concentrations that you use in your experiments?
Oh, yeah. Different laboratories, different dose. Yeah, we reference a paper, Nature Medicine. It is this dose. Some laboratories know it's lower. I'm not sure which one. Maybe I think maybe is correlation the damage never. The damage never use. If it's heavy, maybe high dose, high concentration. Is it nurse maybe low concentration.
But you didn't measure in your rhabdomyolysis model. You haven't.
No.
Okay.
Yeah. Thank you.
Okay. Thank you very much. And we are closing this session. I would like to thank all the speakers as well as the audience for their participation. And that's it. Thank you.